Hello, it's your boy, the one and only Sir Patrick of the Department of Biology. And yes, this is Lecture 3, Taxonomy and Phylogeny of Algae. And before I start our lecture, I'd like to remind you again of the reading material that we have. And I'm about to do the quiz for that right now. So you better watch out for that quiz. It's a good quiz for that reading. So you guys are forced to read the reading material that I uploaded, Patrick Killing. So this is one of that figures. No, this is one of the figures that we had to experience in the reading. It is a very good figure, if I say so myself. And you can see the different supergroups that are there. So the Plantae, the Rhizaria, Unicorns, Excavates, and the Chromalgulates. So as you can see there, no, we have all of those highlighted in black and white letterings. So those are your organism with plastids, including the Plantae, no, the land plants, because they are again closely related to your charophytes, your algae, the green algae right there. And of course, if you are finding a hard time locating us humans, we are here. No? We are here with the unicorns, with your slime molds, your fungi, your fungi, your microsporidiae, the slime molds, and those kinds of things, organisms. So this, I want you to, for us Bio 102 class, I want you to frame this. And I also uploaded you can frame this and laminate it if you want, or use the other tree that I uploaded in our VLE course site, the other reading that I uploaded there. That's a more updated tree of life. But this tree right here is based on, well, it's based on many molecular evidences. It's a concatenated trees from your gene evidences, molecular gene sequences, both your plastid, your mitochondrial, your nuclear genome protein sequences, and your morphological, your biochemical markers too. So this reading from Patrick Killing is a very good example of how we do polybasic taxonomy. So if you remember your Bio 110 class from us, Sir Dane, we talk about polybasic taxonomy, and this is how it looks like. You use every evidence that you can get there, and you produce a tree of phylogeny. For that matter, you group organisms based on many evidences that there is. So yeah, another interesting figure from the reading material is this one. So I'd like to laminate this too if I have the chance. I think that's only 120 pesos for this size. I don't know, from sa session, no? 120 lang ata ito. So ayan, no? Yeah, this is a very good figure too. It's color-coded, as you can see here. So yeah, it's a very good figure. And as you can see, the color stands for something. Yeah, and this one is the weird one, right? Remember the Polynella? It's a very weird one because he exempted it. It's like an exception from the other organisms right there. And as you can see, the organisms right here below, these are the organism that has a very problematic way of acquiring their plastid. Some of them acquire, then lose, then acquire again, the dinoflagellates and the diatoms, which are grounds for controversies in our tree. Yeah, remember what he said. And yeah, so we have the primary plastids, the organism with the primary plastids, your glaucophytes, your red and your green algae, and those organisms, or a version of those organisms, in turn get eaten again through the process of endosymbiosis. They didn't get digested, And so they form some symbiosis with the host, no? So yeah, hindi lang pala isang endosymbiotic event yung nangyari. Madami pala, no? Tapos meron pang mga nagkakaroon ng serial endosymbiosis. At yeah, napaka-weird talaga ng biology kung makikita niyo. Ang ganda ng figure. Color-coded yan sa kingdom Plantae, the excavates, the chromalveolates, yung color pink, di ba? Excavates kasi kakulay din siya dito. Right here, di ba? The yellow, the green, Plantae, supergroup, the excavate supergroups, chromalveolates yung mga light blue. Ayan. So yeah, no? Better watch out that. And again, I'd like to remind you, if you are in my Bio 120 class, microbiology class, we had this portion. Siguro pwede kong i-show yung figure niyan dito. We had this portion that, we have this reading that we talked about the weirdness of the endosymbiotic model. The way an organism engulfs something. And this paper talks about a new model for endosymbiosis, wherein the host doesn't really have the energy to engulf. During those billions of years in the deep time of geology, during the earlier periods of evolution, if you imagine yourself as this organism that engulfs these plastids or the mitochondria, it would be impossible. Well, the paper says that it would be impossible for an organism to generate that energy or create that energy to engulf, no? Through vesical engulfation, no? Mga ganyan. Parang amoeba na nag-engulf. Now, weird daw yun. So, it's more likely that the organism, the pre-Luca, the pre-eukaryotic organisms, so our ancestors, no, our eukaryotic ancestors, it's more possible daw na they entangled, then they enslaved the cyanobacterium or the mitochondria by using meshwork or some extracellular protrusions. So yeah, it's still a developing science. It's still up for debates and it's still up for more hypotheses up to this point. But yeah, the general trend, the general conclusion that our scientists are making now is that there is a single, for the case of your plastids, there is a single ancestor. It's the cyanobacterial ancestor that was eaten. Eaten because, again, grabe na yung motor niya. Again, it was eaten, no? It was eaten via the meshwork pattern. So, yeah. So there's that cyanobacterial, or looks like a cyanobacterial ancestor of the plastid that was eaten. And yeah, that's how they are currently thinking of the endosymbiotic event that happened. And from that single event, the single event of the first endosymbiotic event that happened, there are divergence of these three primary plastid possessors, your green and the red and the glaucophytes. Scientists are thinking that the glaucophytes are the first to diverge out of the tree, and then the other two came later on in life, later on in time. And then again, your green gets eaten, and the green algae is also the predecessor or ancestor of your land plants, so that goes to your land plants right there. And your green algae right here gets eaten again through secondary endosymbiosis, no? Again, giving rise to your secondary plastid individuals, organisms, or groups here. And there's again a serial endosymbiosis for the groups right here below. Okay, what's important here to know is that there is not a single, no, especially for the secondary, these portions right here for the secondary plastid. There is not a single, no, it's not a single endosymbiotic event that happened. So figuring out when did that happen is a really difficult job for scientists, for us scientists, no, na i-wrap around yung heads natin sa mga ganyan. Kasi pwede siyang mag-arise through parallel na evolution. And yung mechanism, although yung mechanism kung paano ma-enslave yung plastid, no, sa pagkain ng host cell is sinasabi nila na merong isang singular mechanism na involved, di ba? Sa kung maririnig yun dun sa paper. But yeah, so again, color-coded yung mga yan para matulungan kayo. Okay? So yeah, it's a very good reading. I like the reading so much. So I read it again for the 5th, 10th, I don't know, times. So yeah, to get refreshed on my previous classes. And yes, without further ado, this is Lecture 3, Taxonomy and Phylogeny of your algae. Don't forget killing. And thank killing myself. Welcome to Lecture 2, Taxonomy and Phylogeny of Algae by yours truly, Sir Patrick James F. Penal. Now, let's first have the outline of our lecture for today. So for our outline, for number 1, we'll have to discuss the 3 fields of systematics. We're going to briefly discuss on the taxonomy, the concept of binomial classification, and we'll browse on phylogeny and evolution. Next, we'll move on to the basis, part 2 of your algal classification. Then we'll move on to the different algal groups that we have here. Okay, so the objectives of our lesson for today would be, by the end of the lecture, the student should have identified and describe, should be able to identify and describe the 3 fields of systematics, to enumerate and understand the basis of algal classification, to investigate the relationship among different algal taxa or algal groups, and to construct a simplified summary of these algal taxa. And lastly, to gain a deeper sense of appreciation for the overall phylogeny and taxonomy of algae. So the lecture for today is, it's essentially a summary of what we have read, the reading material of Patrick Killing on the origins of plastids. Okay, so moving on to our lecture, we have the 3 fields of systematics. We have taxonomy, or the biological nomenclature. And taxonomy really has 1 or 2 goals. Number 1 is one kind of organism should only have one correct name. That's the first goal of taxonomy. And number 2, that no 2 kinds of organism bear the same name. So Carolus Linnaeus, back then in 1758, he coined the term binomen, 2 names, diba? Laging 2 names. And it is from the dead language Latin. We use Latin because it's a dead language, so it's not gonna change anytime soon. And the following rules that we're using here in the taxonomy of your algae should follow or should be consistent with the rules of your ICBN, or your International Code of Botanical Nomenclature. And this ICBN right now is not ICBN anymore. It's actually now called the International Code of Nomenclature for Algae, Fungi, and Plants. If you think about it, the major significance of doing all these rules of biological nomenclature, this binomial nomenclature per se, is well, number one is to avoid confusion. No? Remember, it's not just for our human nature of obsessive compulsiveness. No, it's not just because we have OCD. Because we want science, no? We want science to be reproducible as much as possible. We want reproducibility. So to ease reproducibility, we need to have a common name, no? A single name for a single organism. Right? So we have an ease of reproducibility across the scientific community. So imagine the common name and the local name problem. Imagine your talong. So let's draw the talong right here. And yeah. So imagine talong. So we call talong in the Philippines talong, right? But in the US, it is called eggplant. Because before, the first variety of your talong or your eggplant in the US is really yellowish to white tinge, no? It has a yellowish to white color, and it's a bit smaller than the usual elongated talong that we have. It's like an egg. That's why they call it an eggplant back then, because the first variety during the 17th century in England that was brought to the United States looks like an egg. That's why they call it eggplant. But in the UK, they call it aubergine. No? They call it aubergine. Why did they call it aubergine? Well, the origin of that word aubergine is, I think, from the French, because from the French and to the Spanish part of your Europe. No, it came from the word. It came from the word Alberginia, a Catalan/Persian name. So Alberginia aubergine. No, tapos yeah. The UK just copied the name, and their colony, the US, thought of another name, the eggplant. So from aubergine in the UK to eggplant in the US to talong to any other name of your eggplant. So there's a lot of different inconsistent naming scheme. So that really poses a problem for your scientists. So they need to think of a way. So they need to use this binomial nomenclature that was established way back then as a main mode of your identification. So for ease of reproducibility and to avoid confusion across the scientific community. That's why we use naming system. Okay? So let's put the context in our subject. So for the case of your Chloroplasts, so let's have Chloroplasts. Ayan. So wala ulit akong mouse. Wala ulit akong pen tab kasi naghihiraman lang kami. So pasensya niyo ulit yung sulat ko, although wala naman nagbago, pangit pa rin. So yeah, we have Chloroplasts. So Chloroplasts, ito si Ararusip. So there are different species of Chloroplasts in the wild. So if you think about Chloroplasts, there are different species of them. So if you look at Chloroplasts number 1, Chloroplasts sertularioides, if you look at Chloroplasts sertularioides, the leaf of Chloroplasts sertularioides is circular. So if this is the main stem like thallus, so the leaf of your Chloroplasts sertularioides is a bit circular in nature. So the other Chloroplasts naman here, if you think about Chloroplasts lentifera, if you think about Chloroplasts lentifera, the leaf-like structure naman dito, although it's not a leaf, looks like a leaf, no? It's like a leaf. Looks like this. So, ganyan naman kay Chloroplasts lentifera. But if you look on the leaf-like structure of your Chloroplasts racemosa, ayan. It looks like a grape naman. So, para siyang sa end, at the end of the, yeah, it looks like a grape like this. This looks like a somewhat flattened leaf. This looks like a grape at the end. Okay? Yung, the last one, Chloroplasts peltata, on the other hand. So, let's just write pel, ta, ayan. Ang laki ng talong natin nandito. Ay, medyo maliit pa. So, Chloroplasts peltata, yung sa kanya naman, it looks like something like this. Somewhat a nail or something. So, that's your Chloroplasts peltata, Chloroplasts racemosa, Chloroplasts lentifera, Chloroplasts circularioides. So, the names, the specific epithet actually describes the Chloroplasts species. Okay, so that's why it's useful too, in that sense. Okay, so to avoid confusion between the different species, we use the scientific name that is specific for that certain species, for that certain morphology, in this case. Okay, so that's how useful using the binomial nomenclature is. Okay, so what's binomial classification naman? So this is the process of classifying organisms based on what we call the taxons, or the categories of your organism. So we have, so this taxon is any group. Basically, when you say a taxon, it's a group, no? It's a grouping of organisms that is somewhat sufficiently distinct from one another. We will mention what we call the OTU here. So in your biosystematics, OTU is really important because your operational taxonomic unit is a you need to define your OTU. You need to have an operational definition of your OTU. So in the first portion of your publication, for example, you need to say what your operational taxonomic unit is. You need to have that operational definition to classify your groups of very closely related individuals, no? So this operational taxonomic unit, in a nutshell, is simply the group of organisms that you are currently studying. So yeah, that is your taxon or your OTU, no? And syempre, of course, we have your standard botanical hierarchical classification system here. And according to your scientists, your Tulin 2008, the standard botanical classification that is used in systematics of your plants, your fungi, and your algae should follow this. So it has your phyta, your phylum has a phyta, your class is ending in Pisces, phyta, your order should end in Ales, your family Acee. Of course, we all know this from your Botany 119 taxonomy in all of your previous taxonomy classes. So, of course, the genus and species level changes depending on what genus or species you are studying. But yeah, so phylum, phyta, class Acee, order Ales, Pisces, order Ales, family Acee, okay, genus and species level. So let's have an example so this is the one that we're talking earlier on. This is Chloroplasts racemosa. So that's why it's called racemosa, because it looks like grapes, no? Individual grapes. The thallus, the end, the leaf-like thallus looks like a grape, although it's not a leaf, no? So yeah, so sea grapes, the common name of your Chloroplasts racemosa. So kingdom Plantae, since it also follows your standard botanical classification scheme used in your plants, your division Chlorophyta, your class Ulvophyceae, your order Bryopsidales, your family Chloroplastae, and your genus Chloropa, and then the species level is Racemosa. Okay, so that's for your Chloropa racemosa. Moving on to phylogeny. Now, to recap the discussion on your Bio 110 systematics class on phylogeny, we also need to include here phylogeny and evolution. But of course, when we say phylogeny, it should always have evolution. No? If you remember my lectures, when you say phylogeny, it should have the word evolution next to it. Kumbaga. So, evolution. Okay, so evolution. So understanding the phylogenetic relationship among organisms, plants, your animals, and your algae, fungi, and all of them, if you understand the phylogenetic relationship among these organisms, it will help you understand the evolutionary process fully. Of course, no? And the most important thing that we need to discuss here and to remember is the concept of your monophyly, paraphyly, and your polyphyly. So monophyletic. So these are your what we call clades. Okay. Monophyletic, paraphyletic, and polyphyletic. But in a nutshell, when you say a clade, it is a grouping again, a grouping of your organism. But when you say a monophyletic or a monophyly, when you have monophyly, okay, when you say monophyletic, all the descendants and the common ancestors, the recent common ancestors are included in that grouping. So this one right here, letter A, is a monophyletic grouping. However, when you say a paraphyletic grouping, not all descendants of the common ancestors are included in that grouping. So for example, if this is a is a paraphyletic grouping, okay, this letter B right here, this ABC right here is a paraphyletic grouping. It is a paraphyletic grouping because not all the descendants are included. This taxon right here, letter D, is not included in the clade. Okay? Now, if you say naman, a polyphyletic, the grouping is a polyphyletic grouping, descendants of two or more, the descendants that you are including in that clade, okay, are of two or more ancestors. So, for example, this is a polyphyletic group because the descendants are of came from two or more ancestors. So yeah, let's erase that. So in phylogeny, you try to determine the origin of the species by doing constructions of your different phylogenetic trees, your cladogram, your evolutionary tree. The tree, you are essentially creating an evolutionary dendrogram or an evolutionary tree. Okay, which there are different branching of the trees. The different branching of the trees of your diagram depict the relationship of your organism. So the different branching here depicts the relationship of your organism. Your nodes right here, okay, the nodes right here represents divergence of your organism, while at the base, okay, the base of that organism represents the ancestor. Again, this node right here represents a divergence from the common ancestor. The base right here represents the common ancestor of all of these organisms, all of this extant taxon right here. Okay? So yeah, also sometimes your tree is the tree length, the length sometimes, not all the time, but if you create a tree that is based on some evidence or genetic evidences, molecular evidences usually has the tree that you can produce, the length of the tree has some meaning. Okay, if the organism, for example, say the organism branch off like this earlier and the other organism branch off later on in time, that means something. So sometimes when you create a tree, you have different time of branching. So sometimes when you have enough data, enough sequence data, you can infer when the organism branch off from the tree, or if it is an older taxa or a younger taxa. Okay, so sometimes the length of the tree represents how early or how old the species speciate or diverge from your tree. So there are those things that you need to sometimes um, um, take on, no? And consider. But yeah, so our algae grouping right here from Killing 2004 is of what? What type of phyli? Is it monophyletic, paraphyletic, or a polyphyletic? Again, if you have answered our quiz, you should have identified that your algae grouping is a polyphyletic grouping because, again, if you look at the algae groups right here, here. So if you look at these algae groupings that we have here, the descendants of the algae group, all of them, if you look at them, they are a descendants of two or more ancestors. They are descendants of two or more ancestors. Okay, so the ancestors for apicomplexans are here and dinoflagellates. The ancestor for haptophytes are here, here, here. But of course, if you go back in time long enough, of course, if you go back in time long enough, all of us has a single, all of eukaryotes, no? They are proposing, and that's this current stand of the scientific community, all of us eukaryotes have a single eukaryotic ancestor. For a eukaryote, di ba, kung maalala nyo, we descended from Leica, and your Leica is your last eukaryotic common ancestor. But all of life originated from Luca, or the last universal common ancestor. Well, that's the hypothesis and the theory right now, the scientific community. So yeah, if you go back, way back in time, that dot right there is your Leica, or if you go back way further and include your prokaryotes, can include, and the viruses, I think, yeah, that's your Luca naman, no? The ancestor. But that's the cool thing about your unrooted trees. That's why we use unrooted tree minsan, because sometimes we don't like to infer other things, no? We don't like to infer other things from our tree. And if we just want to illustrate the relatedness of your leaf nodes right here. We just want to illustrate the relatedness between these leaf nodes without making any assumptions about the ancestry. We do not require to root them, to root our tree. Okay, so yeah, you can choose not to root your tree. You can just take that in your mega software, and it can do that. No? You can say I don't want unrooted. Just click on that certain navigation tab, and it will not show roots on your tree. Okay, so yeah, so this is the tree of eukaryotes, no? The diversity of plastid bearing eukaryotes shown with your current black and white letters. So yeah, so we have your different supergroups. We have your plantae, chromalveolates, your rhizaria, excavate, and unicorns. So in these three, these eukaryotes are divided in that 5 large supergroups. Of course, this unrooted hypothetical phylogeny of your eukaryotes is based on many gene trees. So it's based on many characters. That's why it's an example of your polyphasic taxonomy, because we are using different evidences to create this one single tree. We are using protein insertions and deletions. We're using cellular and biochemical markers. we're using morphology, no? Cellular, biochemical markers, and similarities. We are using all of them, plus your gene sequences, your plastid, your host genome protein sequences, all of these evidence just to create that one single concatenated trees. Okay? So yeah, by synthesizing all the data from your individual gene phylogenies, from the different gene trees that we have in our database, and other kinds of your molecular, biochemical, morphological markers, all of this, we can broadly outline the different, we can broadly outline the global phylogenetic tree of eukaryotes. We can outline it like this. Sorry. Ay, ang bagal. Ay, ayan. We can outline it like this. So that's polyphasic taxonomy. We use every bit of evidences that we have, just to make the early reconstruction of your evolutionary tree for the case of eukaryotes. Okay? Yeah. But mostly now we are using the recent methods that we use are more of the molecular data, based on the small subunit SSU rRNA sequences. Yeah, 18S rDNA sequences, yung mga ganon. But the thing that I want you to know is that this is really the real challenges is not to generate the data. The real challenge is to synthesize all of these evidences, to use several evidences and merge all of this to create that one phylogenetic construction that we ought to use. We're done with the basis of algal classification. We have a lot of bases, from your morphological to cellular structures, biochemical similarities, gene sequencing, and the synthesizing all of those evidence together, polyphasic taxonomy. And now we'll move on to the algal groups that we have here. So for the part 3 of our lecture, we'll talk about first the theory of endosymbiosis, the types of chloroplasts that we have, and we'll move on to the algal classification based, of course, on that chloroplast that they have, and the 4 major algal groups. Okay, so again, plastids are organelles containing photosynthetic and other biochemical pathways for compounds. That is very useful for your photosynthetic organism, because plastid, in your plastid is where photosynthesis occurs. And your algae, as we know, algae are your cells that undertake photosynthesis. There are exceptions, of course, and or possess plastids, but also there are exceptions. Sometimes they lose it, others are relict. Okay, so algae are not a group. So your algae groups is not a group like your plastid, your plants and animals. It's not a group like them. Algae are a collection of unrelated organisms that possess this plastid, are sometimes eukaryotic and sometimes are not, and sometimes they are photosynthetic. Most of the time they are photosynthetic. Sometimes they are not photosynthetic. Okay, there are exceptions, what I'm trying to say. So there is endosymbiosis in your algae groups. We need to talk about that. So according to Keeling, if you have read our reading material, so this will go on very smoothly because you have read the reading, okay? And we took a quiz for that reading. So for that reading material, you have seen the endosymbiotic events that happen. We have the primary endosymbiosis, the secondary endosymbiosis. So if you look at this figure right here, this is a figure of your primary endosymbiosis, wherein your heterotrophic eukaryote eats a gram-negative cyanobacterium. Okay, so your heterotrophic here, the eukaryote right here, eats a free-living cyanobacterium. So they are saying that it is a cyanobacterium, or sometimes they are telling us that it's a cyanobacterium line. But yeah, the general consensus of your scientists is that a cyanobacterium or other representative of your cyanobacterium was eaten, okay, by your heterotrophic eukaryote. So the heterotrophic eukaryote eat the cyanobacterium, the gram-negative cyanobacterium, which retained it, retained it in the cell here, retained it in the cell rather than being digested. The cyanobacterial endosymbiont is substantially reduced, and a large number of genes are transferred from the nuclear genome of the host. So that's the thing about DNA exchange. If you talk about the DNA exchange in cyanobacteria, there are a lot of ways for them to acquire or lose their DNA. So your cyanobacteria can perform transformation. So wait lang tayo sa pagsusulat ni Sir. Surprisingly, maganda yung pagsusulat ko ngayon, even if I'm just using a mouse. So transformation. So when we say transformation, it is when your DNA exchange happen from your directly from your environment. So you randomly just take DNA from your environment. So that's transformation, okay? When you say naman to transduction, okay, let's do it again. Transduction. So this is again a type of DNA exchange, but this time instead of from your environment, from your plasmid naman ito. Okay, so plasmid, transformation from your environment, transduction from your plasmid. And the last one that your cyanobacterial can perform is what we call mating, or your cell conjugation, which are pretty rare, if I am to say to myself. This type of mating for your cyanobacteria is pretty rare. But what I want to tell you is that your cyanobacterium here, the cyanobacterium that was eaten here, and the cyanobacterium right here inside the cell during the process of endosymbiosis, can perform gene transfer from the chloroplast to the nucleus. And sometimes there are a lot of papers that I am reading right now that say that the gene transfer from the genome of the endosymbiont to the host nuclear genome is often, and is in big chunks. So it is frequent and in big chunks. So it makes sense that your the nucleoid of your cyanobacteria right here gets reduced when it became the plastid of the host organism, because most of the genome of your endosymbiont gets transferred or gets acquired by the host. The mechanism of this is still to be elucidated, but there are a couple of theories regarding this. So you can read on that. After being eaten by the heterotrophic eukaryote, the cyanobacterium didn't get digested. Rather than being digested, it get to be retained in the cytoplasm of your host heterotrophic eukaryote, and then the large genes of your cyanobacterium, the gram-negative cyanobacterium, gets to be transferred in the nuclear genome of the host. The protein products of these genes, both inside your cyanobacterium and inside inside the plastid and inside the nuclear genome, are protein targeted. So there is the protein products of these genes are targeted to the plastids or to the nucleus by a way of transit peptide, what we call transit peptides. So these are important peptides. So these are like signaling molecule in front of your protein product. So if this is your protein product, the transit peptide usually sits in front of that protein product. So it should first bind to your receptor before it gets to be transported inside of your chloroplast. Because remember, we have multiple boundaries here. We have multiple cell wall of your plastid, because your plastid essentially, no, your plastid is also bounded by your outer and inner membrane, because that was the inner membrane of your cyanobacteria that was eaten earlier, the cyanobacterium. And your phagosomal membrane right here, the phagosomal membrane right there, is pretty important. It's not that important, I mean. That's why sometimes the presumed phagosomal membrane of your host gets to be lost, as well as the peptidoglycan cell wall of your cyanobacterium right here. Because they are not that important in the photosynthetic process, they gets to be lost. Okay? So in the quiz, why did we say that the two membrane of your the inner and the outer membrane of your chloroplast is important in photosynthesis? So because they are in the synthesis of your thylakoid membrane. These membranes are thought to have invaginate inside to create that thylakoid layers right here. So these layers here are thought to invaginate inside to create that thylakoid, that granum grana, that the stromal, the lamellase, no, the stacks of thylakoid that we see. So your primary algae, so this is your, since this organism perform primary endosymbiosis, they are called the primary algae. So your primary algae, their plastid should have should always have at least two. Their plastid should have two membranes, should be bound by double membrane. So their chloroplast or their plastid should have double membrane. Okay, they should always have double membrane. This is the inner and the outer membrane of your earlier cyanobacterium. Okay? It should always have two, because that's the cyanobacterial inner membrane and the cyanobacterial outer membrane, which was eaten. Now for your glaucophytes, it has three because the peptidoglycan did not disappear right away. Okay? So this is your common cyanobacterial cell. It has two membranes. It has your cell wall, of course, peptidoglycan cell wall, the thylakoids, the nucleus, or the nucleoid pala, the nucleoid or the chromosomes is, the circular DNA is right here. Okay? So now, for the case naman of your secondary endosymbiosis, your eukaryotic primary algae, okay, is eaten by another second eukaryote right here, is eaten by this eukaryote right here. But this time, again, just like in your primary endosymbiosis, the endosymbiont, the primary algae, did not get digested. Instead, it got retained, but most of its cytoplasmic components gets to be reduced, gets to be degenerated. So most of the cytoplasmic components right here gets to be reduced into a sorry ass of a chloroplast. Okay, so most of its cellular components, and sometimes its nucleus, and most of the time nga, its nucleus gets to be reduced totally. Instead of having a nucleus, it gets to be transformed into what we call nucleomorphs. So this the nucleus of your primary alga can sometimes be retained, but it is of a highly divergent form. We call this one the nucleomorph. So the nucleomorph is a very reduced nucleus of your primary algal symbiont. Okay, so your eukaryotic, your primary algae that was eaten, most of its genes here again gets to be transferred now, and from the nucleus gets to be transferred to the nucleus of the secondary endosymbiont. And since we now have at least four membranes, you need to target that. But let's first mention what are these membranes. So why 4? Because again, we have the outer membrane of the initial cyanobacterium, the inner membrane of the cyanobacterium, again, of the cyanobacterium. And you should also have here, this one right here, the normal membrane, no? The membrane of the endosymbiont, okay? The membrane of the endosymbiont, that's the third. And the fourth one is the phagosomal membrane. Phagosomal. So it should have at least 4, or at most 4 membranes. You have the inner and outer membrane of your initial initial cyanobacterium. You have the membrane, no, of your endosymbiont, no, the eukaryotic cell right here, the membrane, and the phagosomal membrane of your host, the second host here, which is also a eukaryote, the phagosomal membrane. Sometimes, again, the phagosomal membrane gets to be sometimes your primary algae cytoplasmic membrane gets lost in the, so this one right here, the endosymbiont, the membrane of your endosymbiont right here gets to be reduced. So what is maintained is the phagosomal membrane, the inner and the outer membrane of your initial cyanobacterium. So it's a triple membrane chloroplast. If it's a quadruple membrane chloroplast, no, it should have the outer, the inner of the initial cyanobacterium, the membrane of the eukaryotic primary algae, the endosymbiont right now, and the phagosomal membrane of your new host eukaryotic cell. So just like in your primary endosymbiosis, again, your organism, since it was reduced, no, there will also be a gene transfer, again, from your organism that was eaten to your host nucleus. So there's a gene transfer from this, so let's erase, wag pala. So dito ko na lang ilagay. So there will be a gene transfer from this here, right here, to the new host, and from this to the new host. Again, there are gene transfer that will happen. So due to that, again, your chloroplast nucleoid genome would be reduced, and also the nucleus genome of the primary algae will get reduced. So it will become the sorry-ass nucleomorph, or it will be completely lost in the process of secondary endosymbiosis. But due to that gene transfer that are happening, you need to, of course, do some protein targeting again. So you need to have protein targeting. Targeting. target thing. So you need to have protein targeting. Why? Because again, you have different membranes. If your protein products are produced right here, if your protein products are produced right here, and you need that protein products to enter here, you need to target those proteins. No, you need to have a protein targeting. So remember earlier in our primary endosymbiosis, we have that transit peptide. Right now, we also have that transit. Aside from the transit peptide, we now have the signal peptide. Signal peptide. So these are in front again of your real protein product, the mature protein product, because it needs to be recognized by those different membranes and the different receptors for it to go inside of that plastid or for it to go outside of that plastid. Okay, so it works both ways. So in your bio 150 in your bio 150 in cell biology classes, you will talk about protein targeting. So these are the whys of why you're doing that why you're doing that talk on protein targeting, because you have these membranes. There is this kainan na nangyayari, no? Si crush na kinain ulit. So madaming kainan na nangyayari. That's why we are talking about this protein targeting, the signal peptide, the transit peptide in front of your protein molecule, because it needs to be because it needs to be recognized by the membranes or by the receptors lining that wall, no? That different walls. Ganun kasi, no? Hindi basta-basta papapasukin. These membranes are again selective in nature. So choosy sila. Hindi nila papapasukin basta-basta yung mga protein products na yan. So that's why there's a need for that signal peptide and that transit peptide to target the proteins na palabas kay plastid papunta dun sa host nucleus or papasok kay plastid mula dun sa host nucleus. Okay? So moving on to your five types of chloroplasts, we have number 1, we have a chloroplast with no chloroplast ER. So first pala, before we move on to this discussion, we need to talk about the concept of chloroplast endoplasmic reticulum, the chloroplast ER that we're talking about here. So what is this chloroplast endoplasmic reticulum? So imagine your chloroplast. So this is your cell. If you have a chloroplast right here, so a chloroplast ER is like an endoplasmic reticulum outside of your chloroplast. So it looks something like this. It's a reticulated endoplasmic reticulum. So yeah, it's like an endoplasmic reticulum of your chloroplast. Okay, so these chloroplast ER, again, since it's an ER, it's an endoplasmic reticulum, it has ribosomes attached to the surface of that endoplasmic reticulum. So it has these dots that are your ribosomes attached to the outer surface membrane adjacent to the cytoplasm of your host this cell right here. Okay, this cell right here. So that chloroplast ER right there is studded with ribosome and is close or adjacent to the cytoplasm of the host of the cell itself. So the chloroplast ER is kinda a remnant of your food vacuole, the phagosomal membrane or the plasma membrane. Or sometimes it is referred to as a food vacuole, the phagosomal membrane of the host, and sometimes it is taught as the plasma membrane of the primary alga itself. That's why if you look at this right here, your Chloroplast ER of your chlorophytes and rhodophytes does not exist. Because again, these are your primary algae. No, these are your primary algae. No? And now for this portion right here, they have Chloroplast ER because they ate the primary algae. Remember? This doesn't have a Chloroplast ER because your Chloroplast ER are the remnant of your, are thought to be a remnant of your, either your phagosomal membrane or the plasma membrane itself of the primary algae that was eaten. So remember the four membranes that we're talking about. Yeah. Okay, so yeah, that's your Chloroplast ER. It's the remnant of the food vacuole and or the plasma membrane of the original, no? The membrane involved in the original endosymbiosis leading to the chloroplast in the secondary endosymbiosis. Now you are maybe wondering right here why we don't have, why we don't have cyanophyta, why we don't have cyanophyta as a type of chloroplast. Why don't we include cyanobacteria or cyanophyta here? Well, remember, it was the one, it was the representative of it was the one that was eaten during those earlier endosymbiotic event. So we don't have that type of chloroplast. The cyanobacteria in itself is the chloroplast. Diba? Parang ganun siya. Okay, so we have the 5 types of chloroplasts. We have the first one with no chloroplast ER. Again, we know why it doesn't have a chloroplast ER because it's a primary endosymbiosis. It is a product of your primary endosymbiosis. It has 1 thylakoid per band. So just 1 thylakoid per band. So parang ganyan-ganyan lang siya. Okay, single thylakoid lang. The next type of chloroplast, we have again no chloroplast ER because again it's a primary endosymbiosis. It's a product of primary endosymbiosis. There is no phagosomal membrane. No phagosomal membrane. The phagosomal membrane was lost here. Now, for this type of organism naman, for your chlorophytes, you have at least 2 to 6 thylakoids per band. So parang ganyan. Or 6 na thylakoid per band. Basta 6. Yan, 6. Okay? The next type, these are results of your secondary or the serial endosymbiosis naman. But yeah, most of them are results of your secondary endosymbiosis. Therefore, meron kang, you have your chloroplast ER na. So this one right here has 1 chloroplast ER and 3 thylakoids per band. That's for your dinophytes and euglenophytes. For your cryptophytes, on the other hand, it has 2 chloroplast ER and 2 thylakoids per band. For your primnesiophytes and your heterocontophytes, they have again 2 chloroplast ER and 3 thylakoids per band. So what I need you to think about here is that how much of your membranes, your plastid, no? How many membranes are enclosing your plastids? Okay ba? So for number 1, how many plastid does it have? So let's erase this. Yeah. So how many plastid does number 1? How many membrane is enclosing our plastids here? So we have 2. So for this naman, for the number 2 type of chloroplast, we have again 2. Now we didn't include here your glaucophytes, but your glaucophytes has at least 3. The outer, diba? The glaucophytes, remember from the reading, glaucophytes. So ayan, imagine niyo na lang maganda sulat ni sir. So your glaucophytes have at least 3 membranes. So the outer, the inner, and the peptidoglycan cell wall. Diba? It maintained the peptidoglycan cell wall. Now for this naman, how many membranes are enclosing our chloroplast. So you have one chloroplast ER plus the two initial inner and outer membrane of the cyanobacterium. So you have 3. So here naman, we have 2 chloroplast ER, and for these 2, you have 2 chloroplast ER. So they should have 4. 4 membrane enclosing our chloroplast. So what's the additional 2 right here? The phagosomal membrane of the host, the new host, no? And the membrane of the primary algae. So here, you lost either the membrane of the primary algae or the phagosomal membrane of the new host. So that's why you only have 3. Okay? So yeah, these are the types of your chloroplasts. Now, let's look at them more closely right here. So this is the first one. One thylakoid per band and no chloroplast ER. So again, look at that. So this is the chloroplast, okay? So this is the chloroplast right here. So it has 2 membrane enclosing it, the outer and the inner membrane. And look at the thylakoid, just 1 thylakoid per band. And the ER, the chloroplast ER is non-existent because it's a primary endosymbiosis. So these are for your rhodophytes, your red algae. Their chloroplast looks like this. Now, the next one, we have the 2 to 6 thylakoids per band and no chloroplast ER. So how does it look like naman? So as you can see right here, so again, the chloroplast has no chloroplast ER. So it has no ribosome studding, no ribosomes right outside. And we have 2 to 6, so look at that, 2 to 6 thylakoid per band. So this is your thylakoid band. You could have 2 to 6, at most of 6 thylakoid per band. Okay? This is now naman for your chloroplasts. Now, moving on to the third type, it looks something like this. So again, since a membrane of your, it's either the phagosomal membrane or the membrane of the primary alga was maintained, so you have your chloroplast ER, endoplasmic reticulum. So that's why we call it an endoplasmic reticulum, because again, it is, the chloroplast ER is studded with ribosomes right outside here. Okay? And again, we have 3 thylakoids naman here per band. This is for the case of your dinophytes and your euglyinophytes. Now for the next one, we have 2 thylakoids per band. Again, 2 thylakoids per band and 2 membranes of your chloroplast ER right here. Okay? And here. And we have the another 2 membranes of the chloroplast. The remnant of your, the membrane of the initial cyanobacterium. Remember? So we have 2 membrane chloroplast ER, so 2 membrane chloroplast ER plus the initial 2 membrane of the cyanobacterium. This is the case naman for your cryptophytes. And moving on for the last one, this is naman for your heterocontophytes. We have again 3 thylakoids per band, but now just the same, no? 2 membranes of chloroplast ER, but we have 3 thylakoids per band here. So again, 2 membranes of your chloroplast ER, a remnant of your phagosomal membrane, and the membrane of the primary alga that was eaten, and the 2 membrane of your initial primary initial cyanobacterium that was eaten. Okay? So yeah. So in terms of algal classification based on chloroplasts, we have prokaryotes, the cyanobacteria. We have the eukaryotic algae with chloroplasts surrounded by 2 membranes of chloroplast envelope. we have the eukaryotic algae with chloroplasts surrounded by one membrane of Chloroplast ER. And we have eukaryotic algae with chloroplasts surrounded by two membranes of Chloroplast ER. So we have one. So let's count how many membranes are surrounding the plastid here. So for this, we have 0 membranes surrounding the plastid because there's no plastid at all. The cell itself is the plastid. Remember? The cyanobacterium. That's the cyanobacterium kasi. Okay? So, next is we have this naman, yung pangalawa natin, we have your primary and primary algae, which ate the cyanobacterium and thus have 2 membranes surrounding the chloroplast. So 2 membranes. So that 2 membranes, that 2 membranes surrounding, at least 2 membranes surrounding your chloroplast, that membrane is of the inner and the outer membrane of the initial cyanobacterium. Again, your glaucophytes it's basically the same, but just maintains the peptidoglycan cell wall. Now, for the third classification based on chloroplast naman, here we have your eukaryotic algae with chloroplasts surrounded by one Chloroplast ER. So, in total, we have 3 membranes enclosing our chloroplast. So, that extra 1 membrane that surrounds the chloroplast is either the phagosomal membrane of your host, new host, or the membrane of the primary algae itself. Now, for the last one, we have at least 2 membranes of Chloroplast ER. Now, for this group of organism, it has both it has the initial outer and inner membrane of the cyanobacterium, plus the 2 extra phagosomal membrane of the new host, and the membrane of the primary algae that was eaten through secondary endosymbiosis. Now, what am I talking right here? Ano yung mga pinag-uusapan natin? Bakit may mga ganyan-ganyan pa, sir? Why are we talking about these 4 algal classification? Remember our lesson from killing. So, the first group, the prokaryotes. So, these are this one right here. So, this is your cyanobacteria. So, those are prokaryotic in nature. So, this first group is of the cyanobacterial origin. So, if we look at them right here, this is your 4 major algal groups. We have your cyanophyta. Your cyanophyta has chlorophyll A. They have phycobilisome, phycobilic proteins, your phycocyanin, your phycoerythrin, aside from your chlorophyll A. They have peptidoglycan cell wall, of course, and they also have what we call the myxopycian starch. Okay, so that's for your cyanophyta for our group 1. And this example right here is of the nostoc genus. So this nostoc, again, remember earlier, has from the previous lecture, has your what we call heterocyst. And we'll talk about more of them in the future. They are involved actually in nitrogen and fixation. So that's why they have that. Now, so that's your first group. Now for the second group, we have right here. So this is the second group naman from your killing. Okay? So your second group includes your glaucophytes, your red algae, and your green algae. So let's talk about the glaucophytes first. They are microscopic and usually found on freshwater environment. They represent an intermediate position. They represent an intermediate position in terms of the evolution in endosymbiosis. Okay, so they they represent a intermediate or the transition from your endosymbiont to a plastid. So that's why they are unique in these plastid, because your glaucophytes has retained the initial prokaryotic peptidoglycan cell wall between the inner and the outer membrane, diba? So your glaucophytes are weird in the sense that if this is your chloroplast, you have the outer, you have the inner, but since your glaucophytes are the transition zone, the transition representative, it maintained the peptidoglycan cell wall. Okay, the peptidoglycan cell wall of the initial cyanobacterium. That's why they are thought to be the transition or the intermediate endosymbiotic representative, because they maintain that. Okay, they are also particularly important because they are said to have a pivotal position in terms of the evolution of eukaryotes, the photosynthesis in eukaryotes. So they also have chlorophyll A and phycobiliproteins, phycobilisome. Next is your rhodophytes, your red algae. These are microscopic organisms. Microscopic. These are microscopic and macroscopic organisms. We have your porphyra right here, a very good-looking red algae. Porphyra, it's like a fire, no? Fiery, fiery leaf. And we have Gelidiella. Okay, so yeah, your red algae are microscopic and macroscopic and are present in freshwater, although they are mostly common in marine environment. That's why when you go to La Union and and in Pagudpud, there are a lot of red algae representative, red macroalgae representative. So they're plastid. So yep, they are aquatic, found in aquatic. Their plastid contains chlorophyll A and phycobilisomes again, phycobilins and phycobilisomes. Now, moving on to, hindi pa pala, ayan. So they also contain what we call the Floridian starch. And when usually when your organism has Floridian starch, it means that they are of the red algae origin. Okay, so this is the starch that they have, Floridian starch. Now next, again, still on group 2, we have your chlorophytes. So chlorophytes are the ones that are predominantly found in freshwater environment. So they have chlorophyll A and chlorophyll B. They have starch within chloroplasts. So when you use iodine test in this organism, their plastid will show dark spots, dark to violet spots, because they have the iodine will bind to the starch in their chloroplast. So green algae are roughly divided into the chlorophytes and your charophytes. So there are a lot of green algae in freshwater ecosystem, and your charophytes, the charophyte groups of your chlorophyta is very closely related, or is thought to be the ancestor of your land plants. Remember our quiz, diba? So yeah, so a representative right here that we're gonna mention is Ulva lactuca. So it looks like this in the field. So that's your Ulva lactuca. There's a lot of filamentous chlorophytes in freshwater ecosystem. You might have seen them in your backyard or anywhere else with water. So yeah, that's your chlorophyte. Your chlorophytes are also pesky in nature. They are usually the cause of blooms, and they usually grow too much when there is eutrophication in your environment. Okay, so next group 3 is of this one right here, the euglenids, your dinoflagellates, and your apicomplexans. So that's your group. So first one, we have the euglenophytes. So for your euglenophytes, these are marine or freshwater flagellates. So they have flagella like your euglena and your pecus, no? So again, they have the stigma that we were talking about earlier. So one flagellum with fibrillar hairs or spiral row, and their plastid is of the green algae, diba? They ate, so they are a product of secondary endosymbiosis, and they ate the green algae. So since they have a green algae, their plastid should also have chlorophyll A and chlorophyll B. They have your paramylon. So paramylon is very similar to starch. No? So yeah, this is the polysaccharide that they have. Your euglenids are very closely related to the parasitic trypanosoma, no, that you have I guess you have studied from your bio 111 class, your invertebrates class, your trypanosomes. Nah? So yeah, it's of the euglenozoa group. Okay, so that's your euglenids, your euglenophyta. We also have the next one, your dinophyta, right here. So your dinophyta naman are another large and diverse group of organism which has a red algae derived plastid. They have chlorophyll A and chlorophyll C. So just like euglenid, the major plastid type of your dinoflagellates is weird in that they are distinguished by the carotenoid pigment peridinin, no? Peridinin in their plastid. Again, your group 3 has 3 membrane-bound plastid. Okay? So, your dinophytes are weird, or your dinoflagellates are weird in the sense that they can lose and replace their plastid on several occasions. That's why some representative, no, are weird in that manner. So yeah, your dinophytes are a source of debate and a source of issues in your algae classification. So they also have helical transverse flagellum. If you look at this one right here, you have what we call the sulcus right here. So we have sulcus, or a furrow, in which you have a helical transverse flagellum. It also has a flagellum right here, but it has a flagellum that's what we call the helical transverse flagellum right there in that sulcus. So there's a diversity of diversity of plastid types in your dinophytes because they are the weird ones. Again, next weird one is your apicomplexans. Your apicomplexans are heteroflagellates, heterotrophic flagellates that are composed of groups that are entirely obligate intercellular parasites. A famous example right here is your Plasmodium vivax, Plasmodium falciparum, and those Plasmodiums. Okay, so again, your apicomplexans have red algae-derived plastid, but their plastids are colorless now and are a relic of the plastid that was photosynthetic before. We call this one the apicoplast. So these apicomplexian groups are weird. These are the weirdest organism because they are parasitic in nature, but they are still considered algae. Okay. So there's a lot of considerable debate, and as you have heard in your, as you have read from our reading, Patrick Keeling has made a great deal in mentioning how this group can be a source of considerable debate among us scientists, no? Whether this is really a plastid. Well, it's really a plastid, but the way they are, the way that they became parasitic, and possible implications of this organism, no? Whether it's a green plastid derived or a red plastid, it's still up for debate whether the plastid, your colorless apicoplast, is of a red algae in origin or a green algae in origin. But yeah, that's for them to problematize. Well, next group, for the group 4, we have your cryptomonads and your heterocons. That's for your group 4. So let's talk about your cryptomonads, your cryptophytas. So these are abundant groups of marine and freshwater flagellates. So they contain a red algal plastid with chlorophyll A and chlorophyll C. They are surrounded by 4 membranes, because group 4, again, so 2 chloroplasts ER. In particular, your cryptomonads have nucleomorph, no? With many superficial similarities with that of the chlorarachniophytes. So your cryptomonads have nucleomorph, and they are aquatic in nature, and their plastid is of red algae in origin. Okay, so that's your cryptomonads. Your cryptomonads, again, is host component of the host of the cryptomonad is matter of debate, but is now under the single supergroup, the chromalveolates, remember? But yeah, that's your cryptomonads. An example here is your Rhodomonas salina. Remember, in our earlier lecture, we have the gymnodinium that will eat Rhodomonas salina, no? So yeah, ito na siya, no? Ayan. So yeah, remember the nucleomorph right there. There's your nucleomorph. Baka nakalimutan nyo kung anong itsura niya. Siya yung parang relic na nucleus nung primary algae na kinain. Okay? Na-reduce siya ng na-reduce kasi nagkaroon ng gene transfer, at syempre, ini-enslave na siya nung iyong host na to. Next, moving on, we have we have your Heterocontophyta. So for your Heterocontophyta, your heterocons, these are a diverse group or photosynthetic and sometimes non-photosynthetic groups. So your heterocons has a red algae-derived plastid, and they have ecological significance, no? Like your diatoms and your macroalgae like your kelps. So your heterocons are sometimes photosynthetic or non-photosynthetic. They also have chlorophyll A and C, and most notable for them, they have your what we call fucoxanthin. They have anterior tinsel and posterior whiplash flagella. That's why they are heterocons, because the flagella is of different morphology. So these are the examples of your Heterocontophyta. We have the eight families, at least 8 families under Heterocontophyta. We have your Basiloropyceae, Chrysopyceae, Dictyopyceae, Pelagopyceae, Pleopyceae, and your Primnesiopyceae. Okay? And ayan pa pala. Meron pa tayong Rapicopyceae and Xanthopyceae. So we have a lot of families under Heterocontophyta. So your Dictyopyceae, an example here is your Dictyota. You are maybe wondering where brown algae fits into this story of algae. Pleopyceae is the family for your brown algae. Okay, so we have your Chrysophyceae, Ochromonas right here. We have Basidiophyceae. These are your diatoms. Okay, so your diatoms. So that's why they are called diatoms, because there are like two valves, no? So there are like two valves right here that enclose together. So when we say tomy, diba? When we say tomy, atomy, that's why we call it anatomy, diba? Splitting, yeah, cutting or something. So there are two cuts, something like that, in your diatoms. So these are your diatoms are specifically a microalgae found in main oceans and waterways of the world. So they are the one that cause eutrophic that blooms when there is eutrophic environment. We also have your Pelagophyceae right here. We have here an example of an unidentified ultraplankton, a pelagophyte. There's a very small ultraplankton right here under Pelagophyceae. Chrysophyceae is almost of a golden tinge. That's why it's of a chryso, golden. Chrysalis, remember chrysalis of your butterfly. And moving on to our so yeah, that's your group 4. Maybe wondering why we don't include Polynella in the groupings. Well, because they are the weird, these Polynella are the weird ones. You may be wondering too why we don't include ciliates, because from the lecture of your killing, they are still problematic from the reading material. So yeah, we didn't include haptophytes too, and this right here, and your chlorarachniophytes. But let's look at some of the representative. You have your Gymnochlora stellata. So these are representative of your chlorarachniophytes. It's a relatively rare group of amoeboflagellates. So they also have green plastid-derived chlorophyll A and chlorophyll B. So yeah. So they also have a nucleomorph, a relic nucleomorph, just like your ano nga ba yung may nucleomorph kanina? Just like your cryptophytes, no? So that's your chlorarachniophytes, and this is your Gymnochlora stellata right here as an example. Next, we also have your coccolithopores. These are examples of your haptophytes, no? Or primnesiophytes. So your coccolithoporids are an abundant group of marine flagellates with 4 membrane plastids. So yeah, napakaganda ng mga coccolithopores. I think you have discussed this in your Zoo 111 class. So this is very good electron micrographs of your coccolithopores. Yeah. So these are your coccolithopores right here. These are primary producers in most aquatic environments, and usually their blooms can be large enough and distinct enough that are visible in space. So those are your haptophytes, coccolithoporids. We also have right here your ciliates, although we didn't talk about them and Killing didn't talk about them because their plastid is still up for debate whether if they really have a plastid, no? Kung maalala niyo yung lecture, yung paper. We also have the Polynella chromatopora. So your Polynella chromatopora is weird, no? It's weird. They live and crawl along the bottom of freshwater environments. So they have usually pseudopodia. So they have thread-like pseudopodia. And remember your Polynella is the one that eats your Synacococcus right here. So this is the Polynella. It eats Synechococcus, a species of Synechococcus clade, of a Synechococcus clade. So yeah, it's the one that is weird. Remember. So yeah, it's your Polynella tail is of a different story of endosymbiosis in itself. That's why it's not included in any of our groupings, algal groupings. Yeah, so that's your Polynella that eats the Synechococcus. Synechococcus is a cyanobacterium. No. So if you are very tired of that lecture, if you are tired of using the groupings that we have earlier, this is the one that I'm proposing, and it was also been proposed by Keeling. You can just group them according to what endosymbiosis event happened. So again, your Polynella chromatophora, we cannot group it here in this grouping scheme that we're using. So you can group it to organisms that acquired their plastid to primary endosymbiosis. We have the organism that acquired their plastid through secondary endosymbiosis. We have that organism that acquired their plastid through tertiary and serial secondary endosymbiosis. So we have the glaucophytes, red algae for the primary, and the green algae for the primary endosymbiosis. We have euglenids, chlorarachniophytes. We have haptophytes, heteroconts, and dinoflagellates and apicomplexans for your secondary endosymbiosis. We have some dinoflagellates for the tertiary and secondary and serial secondary endosymbiosis. So if you're tired after a long day of work, just use this grouping: primary endosymbiosis, primary algae, secondary endosymbiosis, secondary algae, or tertiary and serial endosymbiosis. Algae. Now I want to end with this figure right here, because I know we are dealing with a very problematic, very often controversial groupings of your organism that is your algae. If you think about it, it's kind of futile to use the grouping algae because it's polyphyletic in nature. Why use it? It's kind of lost group. Your algae is a lost group. But I find it fascinating and at the same time freeing in studying a wild and free group, a wild and diverse group. And I find a sense of belonging in finding order in a very disordered group like this. And I hope you find that too. So see you next time. This is Sir Patrick. Peace out.